Hidden inside your blood is an evolutionary record shaped by every epidemic your ancestors survived. Thousands of years later, those same biological adaptations may be associated with certain biological patterns related to circulation, aging, and immune response. Every second of your life, an invisible identification system is operating inside your bloodstream with astonishing precision. Trillions of red blood cells travel through your body, carrying tiny molecular markers attached to their surface, almost like microscopic security credentials. In biology, these markers are known as antigens, and they help your immune system distinguish between cells that belong inside the body and potential threats that do not. People with type A blood carry A antigens. Type B carries B antigens. Those with type AB display both, while type O blood carries neither at all. Although these differences appear small, they shape the way your immune system reacts to the world around you. Constantly monitoring this internal network is the immune system itself, functioning like an elite surveillance force. The moment unfamiliar antigens are detected, defensive proteins rapidly mobilize to neutralize what the body perceives as danger. This remarkable protection system helped human populations survive infections and epidemics for thousands of years. Yet, some researchers now believe these same biological traits may also be associated with differences in how the modern body responds to inflammation, cardiovascular stress, and other long-term biological processes. Long before modern medicine existed, the agricultural revolution may have already been reshaping human biology. As early humans abandoned nomadic life and began building permanent settlements, entirely new survival pressures emerged. Large populations suddenly lived crowded together beside domesticated animals, contaminated water, and poor sanitation. These dense environments allowed infectious diseases to spread with unprecedented speed through entire communities. Faced with these constant outbreaks, the immune system likely adapted under intense evolutionary pressure. Some researchers believe type A blood may have become more common during this transition because its immune characteristics could have offered protection against certain bacterial infections common in early farming societies. Unlike type O blood, which carries no A or B antigens, type A blood displays a distinct molecular marker attached to red blood cells that may influence how the immune system responds to threats. These antigens were never just biological labels. They became part of a larger survival strategy shaped by thousands of years of epidemics and environmental stress. Ironically, some of the same immune traits that once protected ancient populations may still influence inflammation and chronic disease risk in modern life today. What once helped humans survive deadly epidemics may now create hidden vulnerabilities inside the modern body. Several long-term cardiovascular studies suggest some population studies have observed differences in cardiovascular-related biological markers in individuals with type A blood, partly because of differences in clotting activity and inflammatory responses within the bloodstream. In ancient agricultural societies, a more aggressive immune profile may have improved survival during bacterial outbreaks spreading through crowded settlements. Modern life exposes the body to a completely different form of stress. Poor sleep, chronic psychological pressure, physical inactivity, and processed diets may contribute to prolonged inflammatory activity in some individuals under chronic stress conditions. Clinical observations have also noted that some studies suggest differences in cortisol response patterns have been observed in individuals with type A blood during stress. Daily pressures such as work deadlines, emotional tension, or constant digital stimulation may trigger stress hormones more intensely and keep them elevated longer. Over time, this persistent strain may influence blood pressure, vascular health, and metabolic function. Blood type does not determine your future, and lifestyle remains the dominant factor in long-term health. Still, these findings offer a fascinating reminder that ancient immune adaptations may continue interacting with modern environmental pressures in surprisingly complex ways today. Across the harsh plains of Central Asia and the freezing regions near the Himalayas, a very different survival strategy was taking shape. Type B blood is believed to have emerged among ancient nomadic populations, constantly moving through changing climates, unfamiliar territories, and unpredictable environments. Survival demanded flexibility rather than heavy biological armor. These migrating communities repeatedly encountered new food sources, bacteria, and infectious threats while traveling enormous distances. Instead of developing an aggressive immune profile like early agricultural societies, The nomadic immune system may have evolved to adapt rapidly to constant environmental change. Over generations, these environmental pressures may have helped shape the adaptive immune traits associated with type B blood. Ongoing research continues examining how type B blood may respond differently to certain infections and gastrointestinal pathogens compared to other blood groups. Scientists are especially interested in how these variations could influence immune responses and inflammation inside the body. Although blood type alone never guarantees protection from disease, the patterns remain scientifically fascinating. Type B blood still appears to carry traces of its ancient origins, reflecting generations of migration, environmental uncertainty, and the remarkable human ability to survive under constantly shifting conditions. An immune system shaped for constant adaptation may also carry hidden weaknesses. In people with type B blood, the same flexible immune traits that once helped nomadic populations survive unfamiliar environments may be associated with different immune response patterns under certain modern environmental conditions. Autoimmune disorders develop when immune defenses mistakenly target healthy tissue instead of harmful invaders. Instead of shutting down after a threat disappears, inflammation may continue circulating through the body for years, gradually damaging surrounding tissue and organs. Medical studies continue examining how blood group antigens may influence these complex immune patterns. Population-level research has also explored possible links between type B blood, and some research has explored potential statistical associations between blood group patterns and certain cancer outcomes, but results remain inconsistent. The exact mechanisms remain unclear, but blood group antigens may influence inflammation, cellular signaling, and how immune surveillance interacts with abnormal cell behavior remains under investigation. Blood type alone never determines who becomes sick. Lifestyle, genetics, metabolic health, and environmental exposure still play far larger roles overall. Even so, these findings reveal how ancient survival adaptations may continue interacting with modern biology in complicated and sometimes unpredictable ways today. Human survival once depended on mobility, endurance, and resistance to infectious disease. During this early stage of evolution, type O blood became the dominant blood group and eventually spread across much of the world. Even today, it remains the most common blood type on Earth. Type O red blood cells carry neither A nor B antigens on their surface, creating a remarkably simple biological profile. This lack of molecular markers may have offered important advantages under ancient environmental pressures. With fewer specialized surface antigens, type O blood appears to maintain a more flexible baseline immune response. Its greatest evolutionary advantage may have appeared in regions devastated by malaria. In regions historically devastated by malaria, scientists observed that infected blood cells often clump together inside blood vessels, increasing the risk of severe complications and organ failure. Some research suggests a reduced tendency for this clumping effect has been observed in type O blood in certain contexts, potentially reducing infection severity in some individuals. Over thousands of years, a potential protective pattern observed in malaria-related research may have helped human populations survive repeated malaria outbreaks, allowing the ghost-like profile of type O blood to persist across generations and remain deeply embedded within modern human biology today. What protected ancient type O populations from malaria may also create weaknesses in the modern world. One important difference involves blood clotting. Clinical studies show that people with type O blood tend to carry lower levels of von Willebrand factor, a protein involved in helping blood clot after injury. Under normal conditions, this reduced clotting tendency may support smoother blood flow and lower risks of certain clot-related cardiovascular problems. During major trauma or severe injury, however, slower clot formation can increase the risk of excessive bleeding compared to some other blood groups. Infectious disease research reveals another evolutionary compromise. Some gastrointestinal viruses interact differently with blood group antigens on the surface of cells. Certain pathogens may attach more easily to cells in people with type O blood, may be associated with differences in susceptibility to specific stomach infections. Blood type alone never guarantees protection or illness. Factors such as lifestyle, immune health, sanitation, nutrition, and medical care still play much larger roles overall. These patterns suggest that survival mechanisms shaped thousands of years ago may still influence modern human biology today. Type AB blood emerged when ancient agricultural populations and nomadic groups gradually mixed across generations. From this biological intersection came one of the rarest major blood types in the modern world. Only a small percentage of the global population carries type AB blood, which combines both A and B antigens on the surface of red blood cells. Carrying both A and B antigens creates one of the most unusual immune profiles found in human biology. Because the immune system regularly encounters both antigens inside the body, it develops a broader level of tolerance toward different blood markers. This allows people with type AB blood to receive transfusions from all major blood groups during emergencies, making them universal recipients. Greater biological tolerance may also involve trade-offs. Some studies suggest type AB blood may show differences in inflammatory and cardiovascular-related biological markers in some studies compared to other blood groups. Researchers are still working to understand how these overlapping immune traits may influence long-term health outcomes. Type AB blood still carries traces of two ancient survival strategies, blending the fortified immune defenses associated with settled civilizations with the adaptable traits linked to highly mobile nomadic populations. Extreme biological tolerance may come with unexpected neurological costs. Long-term observations involving aging populations have revealed possible connections between type AB blood, and some studies have observed differences in long-term cognitive aging patterns among blood groups, though no causal relationship has been established. One major focus involves clotting activity inside the circulatory system. People with type AB blood may be associated with slightly different levels of certain clotting factors, which could influence blood flow in small vessels inside the brain. Subtle changes in circulation and vascular health could gradually affect oxygen delivery to delicate brain tissue. Scientists are still investigating how these vascular changes may influence long-term cognitive health and age-related neurological decline. Blood type alone is never viewed as a direct cause of dementia, but vascular health appears important in brain aging. This creates a fascinating evolutionary trade-off. The biological tolerance that allows type AB individuals to accept all major blood types during transfusions may also interact with clotting behavior and brain circulation in complex ways, revealing how ancient survival traits still influence modern human health. Beyond the major blood groups, another important biological marker quietly shapes immune reactions inside the human body. Known as the AH factor, this positive or negative sign functions like a secondary antigen attached to red blood cells. For most people, it has little effect on daily life, but under certain pregnancy conditions is medically important in pregnancy and transfusion-related medicine. Complications may occur when an AH-negative mother carries an AH-positive baby. Without proper treatment, the mother's immune system can recognize fetal blood cells as foreign and produce antibodies against them. Modern medicine now manages this risk very effectively through preventative clinical care, dramatically reducing complications that once threatened newborn survival. Among all blood combinations, AB negative remains one of the rarest in the world, carried by less than 1% of the global population. People with this blood type face unique transfusion limitations because compatible donor blood can be limited in availability during emergencies due to its rarity. Yet within this extreme rarity lies an extraordinary medical advantage. Because it contains no anti-A or anti-B antibodies, it can often be safely used across multiple blood groups during severe bleeding emergencies. This content is for educational purposes only and is not medical advice. If you found this video helpful, please like and subscribe to support the channel. Thanks for the support.